Welcome to Chapter 4, Functional Anatomy of Prokaryotic and Eukaryotic Cells. Once again, I'm going to be chopping this into two pieces, so this will be Part 1. This chapter fulfills the objective to provide students with the knowledge necessary to comprehend the fundamentals of microbiology, specifically the anatomy of prokaryotic and eukaryotic cells. For this chapter, we're going to cover comparing prokaryotic and eukaryotic cells, and eukaryotic cells. In Part 1, we're going to cover comparing prokaryotic and eukaryotic cells and part of prokaryotic cells. For the second half, we'll finish up prokaryotic cells and move on to eukaryotic cells. For this class, though, since you will be covering eukaryotic cells and lots of other classes, we'll be focusing mainly on prokaryotic cells. Let's start by doing a quick overview of the basic characteristics of prokaryotic and eukaryotic cells. I like to use the analogy of getting a quick overview of the forest before you get distracted by individual trees. Now, from Chapter 1, you may have remembered that prokaryotes are divided into two groups, archaea and bacteria. For the purposes of this chapter, when we say, we meaning me, say prokaryotes, what I'm really meaning is bacteria. For eukaryotes, this includes fungi and protista, plants, and animals including humans. Okay, now for the characteristics of prokaryotes, specifically bacteria. They have no nucleus. The nucleus is a membrane-bound organelle that is the largest organelle in the cell. By organelle, I mean little organs that are inside the cell. They're not really organs because organs are composed of multiple cells, but you get the idea. A nucleus protects the DNA or the chromosome of the cell. Bacteria don't have this. They do have a circular chromosome. They don't have any histones. Histones are proteins that are used to wind up the chromosome, and there are no membrane-enclosed organelles. So not only do they not have a nucleus, they don't have the other membrane-enclosed organelles. Their cell walls are made up of peptidoglycan, you may remember that from Chapter 1, and they divide or reproduce asexually by binary fission. Now for eukaryotes. All eukaryotes have a nucleus. That's what divides the eukaryotes from the prokaryotes. Most of them have a linear chromosome. That means there's two ends. They have proteins called histones that are used to wind up the chromosomes. They have membrane-enclosed organelles. If they do have a cell wall, they never have peptidoglycan. And when the cells divide asexually, they divide either the nucleus by the process of either mitosis or meiosis. Now we're going to go over some of this in more detail as we discuss the different parts of the cells. But once again, this is the forest before we get distracted by the individual trees. In this next section, we're going to talk about the size, shape, and arrangement of bacterial cells, structures external to the cell wall, and structures internal to the cell wall. On this slide, I've listed a bunch of Latin terms that are used to describe the shape and arrangement of bacterial cells. You're going to have to memorize them, I'm sorry. As we go through the following slides, I'll be using these terms, so hopefully that'll help you get a handle on the vocabulary. But these are vocabulary terms that you're going to need to learn. Let's start with the cocci, or the round bacteria. Now, when you divide a circle, you can do so either this way, and then this way, or this way, this way, this way. It all depends upon the bacterial species. Now, some cells, some round cocci cells, once they divide, they have a tendency to separate and they become single cocci. Others have a tendency to still hold hands, and the degree to which they remain attached affects their arrangement. Now, up here, you'll see these are diplococci. They have a tendency to stay together in pairs. So when you look under the microscope, you'll see them in pairs mainly. Others hang on for longer, and these are the streptococci. When you get strep throat, it is from a species of bacterium that tends to stay together in chains, or strep. Now, others have a tendency to form these little sets of four, these flat little squares. We call these tetrads. They're quite rare, but in lab you'll be seeing at least one species that tends to form tetrads. They're kind of fun. Now, if you form a cube, these little guys, we call these sarcina. Now, if you get clusters, grape-like clusters, this is Staphylo, specifically Staphylococci. If you get a staph infection, it's generally caused by a species of bacterium that tends to form these grape-like clusters. Now, when you're in lab and you're looking under the microscope, have a tendency to want to call clusters of rods Staphylo. A rod is not shaped like a grape. So Staphylo only applies to cocci. Remember that one. Now let's talk about the rod-shaped bacteria. These are bacilli, bacilli being the plural. Now, rods, generally speaking, can only divide this direction. There are a few bacteria that can divide this direction, but we're not going to be looking at any of them in lab. Now, if you have just a single bacilli or bacillus, we call that single bacilli. If they have a tendency to hang on for a little bit, they can be diplobacilli. You can also get streptobacilli. No staphylobacilli. Remember that. Now, down here at the bottom, we have kind of a lumpy, short little rod, little bacilli. Now, when you look at these under the microscope, if they're long enough that they're just getting ready to divide, they look like short little rods. If they've just divided, they look like cocci. Now, this confused microbiologists for years, and we had a tendency to call them coccobacilli, or a combination of round and rod-shaped. But what's happening is they are these short, lumpy little rods, as you can see from the electron micrograph. Now, E. coli is a coccobacilli. So don't let that confuse you. If you happen at some point in looking at your E. coli to see all cocci, and other points to see all short little rods, or at other times a mix of the two. That doesn't mean that you've contaminated your culture, that you have a non-pure culture. It just means that E. coli is annoying. It's a coccobacilli. Once rods or bacilli get long enough, depending upon the bacterial species, they can start doing some fun things. For example, they can curve. Now, up here at the top, we have a type of bacterium that tends to form short curved rods that look like commas. We call this Vibrio. Cholera is an example of a Vibrio. Now, if they get even longer, they can form these spirals or helical shapes. Now, they come in two kinds. If they are rigid and they have flagella usually coming out of each end, then they are spirillum. If they are flexible and usually longer and they don't have any visible flagella sticking out of the end, they are spirochetes.